Let's see the third part of the question. Enzymes are biological catalysts that increase the rate of chemical reaction in the body. Which of the following statements are true for the enzyme? So they are only found inside cells, which is wrong because they can be found in non-living cells also. They lower the activation energy of a chemical reaction. Yeah, that is the main function of the enzyme, which acts as a catalyst. Increase the rate of chemical reaction by lowering the activation energy. So this is second part is correct. Third one consists of carbon, hydrogen, and oxygen atoms only, which is wrong. Why? Because enzyme is a protein which consists of nitrogen also, not only these parts. Since they have said only, we have to consider it as a wrong statement. Else we can consider it as correct. So for this part, the only option is 2 only, so which is A. So just mark cross in A. So let's go to the second part of the question. Some chemical inhibit enzyme reactions. The graph shows the effect of the substrate concentration on the rate of reaction of an enzyme. It also shows the effect of two types of inhibitors. You know, type 1 inhibitor, type 2 inhibitors are already shown. But in general case, we have two types of inhibitors as competitive inhibitors and the non-competitive inhibitors. So let's move on to the question. Describe the two differences in effects of these two inhibitors on the rate of the reaction. So for that, what you can write is, the type 2 inhibitor slows down the rate of reaction more than the other two inhibitors, right? So it slows down more than the other two inhibitors, which are type 1 and no inhibitors also. We can consider that. Type 2 causes the rate of reaction to level off, but type 1 doesn't. So these are the two. So here you can see it is going as level off, but in type 1 it is still going on. Okay, so those 2 points can be written in the answer. So let us write that answer first. The first one is type 2 inhibitor slows down the rate of reaction more than the other two. So that's the first point you can write. The next point is what? Type 2 inhibitors causes the rate of reaction to level off. But type 1 doesn't. So these two are the main differences that you can write. So let's go to the second part of the question. The diagram shows the enzyme, its substrate, and a type 1 inhibitor. So you have given a diagram of an enzyme and it has the active site, and then you have two substances like a substrate and a type 1 inhibitor, which can be found, which can be bind onto the active site of the enzyme. So explain how type 1 inhibitor affects the rate of this reaction. Use this information to support your answer. As you can see, the shape of the substrate, this part of the substrate, and this part, which is the binding part. Okay, binding part with the active site are similar. So in this picture, you can see that both are similar, right? The shape of the substrate. And both can compete with each other. That's why this type of inhibitor, type 1 inhibitor, is known as the competitive inhibitor. So to explain, let's just explain that. So explain how a type 1 inhibitor affects the rate of this reaction, use the information. So this method is known as what? Competitive inhibition. And here the shape of the substrate and the inhibitor are similar. Then what we can write? Inhibitor can bind to what? The active site. So since it's binding to the active site of an enzyme, the number of active sites available for the substrate to bind will decline. So the number of active sites available for the substrate will decline. And finally, what will happen is that the rate of reaction will decline. Normally inhibitors will reduce the rate of reaction. And with that case, you have to explain how it is reducing. So this type of inhibitor is known as competitive because it competes for the substrate. The shape of the substrate is similar, that's why it can also bind and competes with it. So inhibitor can bind the active site of an enzyme. So it can bind to the active site of the enzyme. Don't mention like just enzyme, bind to the enzyme. No, it's an active site of an enzyme. So the number of active sites available for the substrate will decline. So it will compete and it will get all the active sites, so the number of active sites that is necessary for the substrate will decline. Thus, what will happen to the rate of reaction? It will also decline. So these points you have to write. So let's move on to the next question. So the first diagram shows the enzyme, its substrate, and the type 2 inhibitors. So this shows a type 2 inhibitors. You can see the shapes are different, right? In this part, this is the shape of the binding part. So type 2 inhibitors, the shape is different from the substrate and it will not bind to the active site. This is the active site. It will not bind the active site. It will bind the part of the enzyme other than the active sites. Explain how a type 2 inhibitor affects the rate of the reaction. So you can write this is what? This is not a competitive inhibitor. This is non-competitive inhibition. So you can say this scenario is termed as non-competitive inhibition, right? So here the type 2 inhibitor can bind to part of the enzyme rather than the active site. So by binding to the other part of the enzyme other than the active site, what will happen? It will change the shape of the active site. So resulting in fewer active sites available for the substrate to bind. Thus, reduces the rate of reaction. You can even mention that since it changed the shape of the active sites, the substrate and the active site will not become complementary to each other. Thus, rate of reaction will decline also. So you can term this scenario is termed as non-competitive inhibition, and it can bind to the part of the enzyme other than the active site. It will change the shape, non-competitive, it will change the shape of the active site, not directly but indirectly it is affecting the shape, resulting in fewer active sites available for the substrate to bind, thus reduces the rate of reaction. So this is the answer that you can actually write for this question. So that's all about the question number 3.